So our next speaker, we move to Plasmodium to plant biology. Professor V Shivaprasad. He is from NCBS, Bengaluru. He is interested in the rare plant, and particularly his interest in the epigenetics and mRNA of those rare plants. He is currently professor and dean of research at NCBS.
Thank you for the kind introduction. I would also like to thank the Academy and the organizers for this wonderful opportunity. So good evening, everyone. My name is Shivaprasad, and I can be contacted in this email. All those plants, I don't call them plants. These are my employers. They make us to work, and they also give us money, make us get money. So our favorite model system is rice, which is here, and its wild relatives. So what we do with these plants, we look at signaling molecules called small RNA. So genome is a recipe book, and one of the recipe that comes out is small RNAs of different sizes, which are made from all over the genome, both coding and non-coding. Everywhere you can get hundreds, thousands of them. And these small RNAs can have 3 different fates, and one of them is a very unusual twist in plants. Typically, when they get into organoid proteins, they can go and target any RNA which shows this complementarity. So it looks very boring. You make small RNA with spending a lot of energy, go and cut a lot of other mRNAs so that the recipe, the protein is not made. But this is a major signaling event which happens in all living organisms. In animals, there is a twist. It goes and the small RNAs goes and bind to the 3' untranslated region. Somehow plant mRNAs don't have long 3' UTRs. This leads to translational silencing, which is very rare to see in plants. But plants have that twist which I told. Certain size small RNAs with certain organoid proteins can go and induce transcriptional silencing, that is epigenetic silencing, by inducing DNA methylation and histone modification. The small RNA-mediated DNA methylation is specific to plants, and it is called RNA-directed DNA methylation. What is interesting is, unlike animals, our germ cells are pre-decided before we are born. Plants can make flower from anywhere. They make these, whatever the cues they see, they can go and make these epigenetic changes, and if there is a flower coming from that region, this will be inherited partly to the next generation. So they pass the message to the next generation. So this machinery is extremely interesting, and this connects to chromatin changes. So in the last 5 years or so, we have looked at how small RNAs are made, what do they do, what kind of organoids are present, what kind of processes they mediate. But more recently, we look at these epigenetic changes, who keeps this message, who passes this message, and so on. So we also look at certain stresses which are also plants sit and manage everything unlike us. So we look at some of them, and I'm going to highlight 2 of these papers first. Harshit's paper showed that how plants can manage injury in no time. So if you cut a dicot leaf, the other part of the leaf is likely to die. But you all see when you cut a lawn, next day the grass pops up. How is that grasses can have this amazing property to grow? So what we find is that plants such as grasses can make tiny peptides within 8 minutes after cutting them. And this induces growth promoting growth hormones, which are 5 amino acid long. And this kicks the growth. So most of the time within 8 minutes, I think we'll be still crying what happened to us. But plants know they promote growth probably because they have been looking at herbivores for millions of years. So Avik's paper which came last year, it looks at regulators of genome imprinting. So genome imprinting in animals is found in different tissues, but in plants is restricted to endosperm. So endosperm is a very unusual tissue. It's a polyploid tissue where I always joke. So most of the time in humans, mother takes care of all the food needs, not just for baby, for everyone. But plants are more general neutral. The nutritive tissue, endosperm, it has 1 part genome coming from father, 2 parts coming from mother, and that means there is a now conflict which gene should be expressed, and this is mediated by a process called genome imprinting. We show who in rice endosperm manages such decision making, which gene should be expressed from whom. So today's talk I'll be talking about Vivek, who is a fantastic student. You can see why. and his recent paper which came a couple of months ago. So the genome, which is the recipe book, is just like any recipe book. Some regions one can read all the time to make the recipe, but some regions which are stained darkly, so each one is a nucleus here. They're all packed in such a way that they cannot be read to make the right protein. And this has many different chemical changes in the right location so that you can either read it or not read it. This decision making so that you can make such a structure is mediated by histone modifications, which are linked to small RNAs in plants. Then DNA methylation is the most primal, most important epigenetic change. What we see is that in the last couple of years, there are these histone variants which can also act as like Capo. So if you see Godfather movie, Capo is the ultimate godfather who looks after everything, right? Good and bad. So same way, histone variants seem to be doing that, regulating small RNAs, DNA methylation, as well as histone modifications. So if you look at the nucleosome, which will be this octamer around which DNA is wrapped around, 147 base pair, and so on, and the tails are protruding out. These are amenable for tiny chemical modifications in the right amino acids. So those are all shown here. So these are amino acids. These are all the different marks. Certain marks help the recipe to be read easily. Certain ones keep the recipe book closed. So in H4, you can see K4 acetylation is an active mark which can help with genes to express, and so on. So these histones, along with these histones, there is paralogs of histones called histone variants. So these are really masters because unlike canonical histones which decorate throughout the chromosome, variants, they go and they are very specific to certain regions and they manage thousands of genes in one go. For example, if you look at SENA3 in animals, it has different names. It binds to the centromere and it is responsible for segregation. You can have DNA damage specific histone variant which comes to very specific region to manage what to do after DNA damage. Such paralogs are well known for H2A, H2B, H3, but not for H4. So whenever a variant is present, it will be simply having a different tail so that you can have different amino acids. You need not have the right modifications or you can allow newer modifications. And these changes are the ones which provide functional specificity. Whenever such variants are present, it can be, let's say, a few hundred genes. They have very nice pattern of decoration. So please see H2AZ here. In the first exon, it decorates, and these will be genes which will come up only under heat stress across plants. And in animals also is conserved, but it slightly does different jobs. So what do histone variants do? How do they work? So Kurmizaka has some answer. If at all somebody can have answer, it's Kurmizaka because he has done some fantastic work. What his work is shows that if you incorporate histone variants where you need a chaperone to come and replace a histone, canonical histone, it can alter the nucleosome positioning, which is like making makkhan, butter churning. So simply alter or give the DNA flexibility or simply throw out or bring a new nucleosome. So what we find is that we have a new histone H4 variant. So H4 is here. So this is the DNA binding domain. This is the N-terminal tail. All those circles, they indicate post-translational modification, which are the ones which give functions for histone H4. In the H4 variant, only the tail is different. Rest of the region is almost identical. You can see many of those where PTMs should happen, they are replaced by different amino acids. The most of the variation is typically in the first 30 amino acids. That's the N-terminal tail. The protein is present only in ORESA members, not anywhere else so far. And if you fuse its promoter with GUS, GFP, we can see it's going to nicely to the nucleus. But what strikes us is canonical histone expresses everywhere. The variant comes in two places, one in the vegetative stage, that's leaf, and in endosperm tissue. So we could make a new antibody for this variant simply because the tail is very different. And when we look into the nucleus, each of these circles are nucleus, we can see it's in the region which should not be read, that's the pericentromeric region, as well as in the genic region, when we performed a ChIP-seq, chromatin immunoprecipitation followed by sequencing, we see quite a bit of binding to the genic region, 1/3 into the non-genic region where you don't have genes. So the peaks are very sharp, indicating that in the beads of string structure, only one nucleosome can have this variant coming, not it is spread everywhere. And such a structure, to find out what are the functions, we took 3 different approaches. We took structural approach, we took biochemical approach, we took functional approach. So I'm going to tell you in the next few slides our baby steps to understand this new histone. What we first did was we purified all these histones from rice, which has been quite difficult to do because H2B in each plant is different. So almost all H2B in animals are almost identical, but it seems like each plant has its own recipe for H2B. And when we managed to make this and also get do the serial dialysis, get the octamers, did use the Vidam DNA, which is typical to go and make canonical as well as histone-containing nucleosome core particles. The size of these are much bigger, you can see. That's because of larger 2B. The human nucleosome, it took only 1 week for us to assemble, but the plant one took years for us. Okay, what we did is we got the cryo-EM structures. It seems to be when it was in bioarchive, it was the first plant chromatin structure. On the top panel, we have the structure for the canonical nucleosome in rice. The bottom one is the variant containing nucleosome core particle. What struck us is that when we have the variant, there seems to be the region is much tightly packed. DNA is wrapped around much more. We got certain amino acids which are different and which could be the reason. So with this information, we started doing some biochemistry. So we first added nucleosome core particle. On the side, we added extra DNA, did a micrococal nucleus which should now trim this nucleosome, did the sequencing, and tried to see what kind of DNA each nucleosome is binding. What we find is that when we have the variant containing nucleosome, the DNA bound by this nucleosome is longer, not 147, 150, but another 10, 20 longer, which you can see here. The DNA entry region as well as exit region have much longer DNA binding when we have variant containing nucleosome core particle. So one of the reasons we found was on the surface there is much more positive charge when you have the variant, so this can facilitate much better binding. So we made certain mutants by incorporating these observations. So another thing, when we started purifying this nucleosome core particle, we found that the variant containing nucleosome are much shorter. They eluted later. You can see in different kind of columns. So it's much more compact. It can hold much more DNA. So that means it is already very easy in the bits on string structure, something which any protein can recognize easily. So when we looked at whether the tail has a specific function, so we took the tails, we labeled with the biotinylated biotin, and did a peptide pull down assay. When we did that, we found that variant containing peptides, it lacked the ability to go and interact with its own nucleosome. So the intranucleosome interaction is curtailed whenever you have the variant containing nucleosome. So one thing which is known for other histone variants like H2AC is that the thermostability of variant containing nucleosome is different. Usually it is feeble, it unwraps much quickly. So when we started looking at whether this is true for the H4 variant as well, the thermal decay profile is shown here. So the canonical one, it is dissociating at 87 degree. The variant is at least 5 degree lower. So when we took certain amino acid mutations based on our structural prediction, we could recover even with single amino acid mutation half of its thermostability. All these variations, whether you have less stable nucleosomes, so whichever way, they are favorable spots for chromatin remodelers to go and organize the genome in different way, what to read and what not to read. One thing which we wanted still to check is whether in a octamer you can have two proteins of the same type. You can have variant in the octamer, or variant or canonical together. So these are called as homotypic and heterotypic nucleosomes. So the pictures, they clearly show better than what I can say. So if you have variant, means both are variants. If it is heterotypic, means one is variant, one is canonical. To see which of this is true, we took in vitro and in vivo approach. So maybe I'll skip to the in vivo approach. Here we raised transgenic lines with the variant containing flag tag with the variant and did micrococcal nucleus assay. And we did this immunoblotting for the tails. And what we see is that wherever you see the variant, you have only variant. Wherever you see the canonical, you can see only canonical. That means H4V containing nucleosomes are homotypic. Now we wanted to look at their functions. So the only way is obviously to look at the knockouts. So we raised CRISPR transgenic lines in rice. So we got single copy transgenic lines. And after 3 generations, which typically takes 3.5 years, backbaking for most of the PhD students. So we can remove the Cas9 and still have the edited line. And this we could get these plants. And when we looked into these plants, we got 2 surprising phenotypes. One, in the seedling stage, I said previously, the protein is expressing in the seedling stage, leaf stage, and endosperm. We could see the plants are sick. They grow much less. Their root length is same, but you can see the aerial part is not growing well. And when we raised transgenic lines, we could see the seeds are much shorter. So if we have a complete knockout, there was no seed filling. So we got 2 different phenotypes. Steffi is looking at what exactly this is made up of, but I'm going to concentrate on this phenotype. So we performed RNA-seq. When we performed an RNA-seq, we could see that many of the genes which are changing, they're all well known for stress response in plants. So typically, they're all of this category. All these are well-known stress-responsive genes. Basically, the plants are stressed. They mimic some kind of a stress. What kind of a stress it can be. So we did a PCA with all the stress data sets that we and others have generated, which is around 320 for indica rice. And we started comparing what kind of differentially expressing genes are seen in the knockout plant. When we performed this analysis, surprisingly, the closest, it's almost overlapped with salt stress, but not any other stress. So plant people always think that salt stress, osmotic stress, drought, they're all connected. But this showed that it's connected for 5-10 genes, but not thousands of genes. So many of those genes which should come up only after salt stress, they were already up in the knockout without having any salt stress. So here in the scatter plot, you can see there is almost a near perfect overlap between DGs in knockout versus genes which are coming up only after salt treatment. So what happens to the plant now? Plant is already feeling like stress. If you even more give more stress, what happens? Nothing happens because plant is already feeling like it is under stress. It did nothing because there is nothing else it could do. So what is happening under salt stress? So under salt stress, we could see there is a relocalization of the protein to more genic regions. There is a reorganization at the global level. The peaks became wider, not just one nucleosome. It could spread to several other nucleosomes in the vicinity. And there was enhanced binding of this variant. Wherever there was binding of the variant, we could see the most common overlapping signal is H4K5 acetylation, which is an active mark for stress. In the knockout plants, you can see there is nothing happening. So this variant, whenever it is present, it is the one which is promoting active marks of H4K5 acetylation. Okay, so if you look at all the stress response, one set of the stress response genes are under H4 variant. So this is roughly around 35%. Only those are under H4, but not the others. So it is very specific only for salt stress. So what we show here is that in the normal condition, without any salt stress, there are regions where H4V, which is shown in green tail, is binding and suppressing, not allowing the expression. But when there is a stress, this canonical comes back. canonical is always filled with the active mark. So it is preloaded with the H4K5Ac deletion mark. This is something a fantastic Shiv Grewal has identified this couple of years ago in East. So whenever it's replaced and brought back with the canonical, the genes can now change expression. So the way the homotypic compact unstable nucleosome exists, it can be easily read so that you can replace this region. The result is that plants can now cope with salt stress. So this came a couple of months ago. Why rice, or why in aquatic plants? So rice evolved from such semi-aquatic weeds. Hardly any grains are made in those. From this, we converted during domestication these fantastic plants which can have a lot of grains. Now, even in the agriculture system, sometimes from morning to evening, there is increase in salt. In the end of the day, you are likely to have more salt. Even within the season, there are different growth stages where the salt can really increase. So it seems like plants have, those are coming from aquatic background, they seem to be having this way to act quickly without really damaging too much themselves. So where does this work fit into what we know about salt stress? People knew how to see what happens when there is a salt stress, what kind of genes go up or down in the nucleus, but why, it was not known. What we show is that this variant is the one which acts as a gatekeeper for all such genes, so that 1,300 genes, they can change at once. So I would like to thank my lab. So the work I mentioned comes from a fantastic PhD student called Vivek Harisundar Gandhivel. So these are the people who have passed out. So this is my current lab. David is the real discoverer of small RNA, and Weltham is my PhD supervisor. These are my source of inspiration. These are my fantastic collaborators, JP and Vinod. They are the ones who helped us with the cryo-EM structure. So we made the nucleosome preparation, but getting the structure was not with us. These are our funding agencies, and thank you for listening. So I just have one more slide. So other than this, I do a lot of outreach talks, roughly many per month. Typically going to schools, so you can see in this school, students are sitting on the floor, but they are attentive, which is quite important. Only those who are not attentive are the teachers, who are probably doing some gossip. Second thing I do is I do write poetry on science and scientist life in Kannada, but this is also translated in English. If anybody want to see at least e-copy, I'll be happy to share. Thank you for listening.
Okay. Fascinating talk. And of course, I mean, your science communication skill also great. It's very important. So.
Hi, very nice talk. So I was curious that you showed that with this variant mutant, there was no effect on the roots. But with salt stress, in one of the schematics, you were showing longer roots, right? So I was wondering if you have any insight on alternate mechanisms and why this is not, is H4 variant not in the roots?
Very good observation. So one of the transcription factor is the one which meddling with the root. Typically, if you put rice plants in root stress, the roots go longer. Okay? So the roots in the seedlings didn't go because it was taken early. Later, you can see there is one transcription factor which we know which can promote this longer root. But the plants already show, then they don't do anything. They are already feeling it's already stress. So, yeah.
Hi. The target genes for H4V that you said that mostly they are the stress responsive genes. You found that their expression has been changed. So is it like they are the direct target of on the chromatin sides? These are the genes that are targeted, or it is possible that there can be some secondary effect of that?
Yeah, thank you. So we looked at detail. 104 genes are direct targets, and out of that 60 are transcription factors. Okay. So their downstream descendants are the ones which make up 1,300 genes together. And so we could map literally each gene, track each gene.
Got it. Yeah, and is there any correlation between facultative and constitutive heterochromatinization with localization of this H4V new variants?
Yes, there is. Initially, without salt, it mostly resides in the heterochromatic region. But under salt, now it goes and also shuts down genes which should not be up during salt stress. So, so it does both. It's not just activating genes, it also has to suppress genes.
Thank you.
So if you look at different varieties of rice or different species of rice, you find that their ability to tolerate stress also varies. In that case, what happens to this histone?
Excellent question. So we have a salt-tolerant variety called Pokali, so which grows in Kuttanad area in Kerala, and it is naturally salt-tolerant. We see that distribution is very different. It is already half active. So the variant works very differently. Variant has one extra amino acid change compared to all other 3 serves and rice. So in Pokali, we work on Pokali as well. And we see it's like it's half changed, but without compromising its yield. So it has evolved in an environment where it has, plants are, I always say plants are too clever just because they are ancient. And they stay in one place. They can't escape. So they have nice way of using this variant as a switch to go and change a little bit, but not compromise the yield.
A non-specialist question. Of late, there is a lot of anti-propaganda about rice. Is there something one can do to the genes to make it healthier?
Thank you. That's what we do. What we call it is we are trying to do de-domestication. The domestication, the last rice which we are eating is green revolution lines, which are created when we are dying of hunger, literally. So we have created green revolution lines, and all of them make a lot of starch, but nothing else. But our endogenous lines, which are native adapted, these are called land races. I mentioned about Pokali. These don't have that profile. They are actually good. So I know we all scold or we say carb is bad and so on, but carb is very important as well. And it's not like our ancestors were always doing something or the other. If you see old pictures, they're all sitting long time. It's nothing to do with that. I think what we have done is we have taken green revolution lines which make a lot of only starch, but nothing else. And the way we eat, we mill and remove all the important things, and we eat pure starch. So if we take unpolished rice, we get quite a bit of nutrients. If we go back to land races, which many of you seniors, we all have grown up eating them, they're all still fantastic. They have many nutrients beyond starch. So what we can do is, or we are trying is, bring back some of these qualities from land races back to rice without compromising yield, because we can't allow reduction in yield, then it will be quite difficult to manage.
Okay, thank you, Professor Shivaprasad.